Hello students, so today I am going to discuss the Karnataka CET Zoology paper. So let us start with question number 1. Which of the following is abbreviated as ZIFT? So here the ZIFT stands for what? Zygote intrafallopian tube transfer, right? So you can see the option number 1, zygote intrafallopian tube is given, transfer is not given. Zygote interfallopian tube, no. Option number B, zygote intrafallopian tube transfer. So answer should be second one. Answer should be B. Fourth option also you can see zygote interfallopian. There is not inter, it is intra, right? So answer should be B. Next, an example of hormone releasing IUD. IUD means what? Intrauterine device, right? So you can see Lippis loop. Lippis loop is what? Unmedicated one, okay? So it is not hormone releasing. Implant. Implant is not an IUD. Multilode 375 is a copper releasing IUD, whereas LNG20, levonorgestrel 20, that is progesterone releasing or hormone releasing IUD. So answer here will be B. Next, MTP are considered relatively safe during. MTP means medical termination of pregnancy, which is considered generally safe in first trimester within 12 weeks. Okay. So answer is third one. It is direct question. First trimester within 12 weeks. Next, from the following table, select the option that is correctly characterize the various phases of menstrual cycle. So, menstrual cycle, different phases it has been mentioned like menstruation phase, follicular phase and luteal phase. Now we know that during the menstrual phase what happens? Menstruation will occur or menses will occur. So answer is either A or D. So straightway eliminate B and C. So mature follicle is not formed during menstruation phase, or regeneration of the endometrium will not occur in menstruation phase. So either answer is A or D. So now let us check follicular phase. So during the follicular phase what happens? You can see the LH surge. LH surge will occur on the around 24 hours before ovulation, right? So on the 13th day approximately LH surge will occur. So follicular phase is correct here. Developing corpus luteum. Corpus luteum is not present in follicular phase because after ovulation, what happens? Corpus luteum will rupture and secondary oocyte will be released to the fallopian tube. Then only this empty Graafian follicle will be transformed into corpus luteum, that will occur in luteal phase. So here it is wrong. Also, during luteal phase, they have given follicle maturation. Follicle maturation will occur in follicular phase, not in luteal phase. Luteal phase, corpus luteum will be developed. So it is also wrong. So here answer will be first one. Coming to the next, and yes, in luteal phase, regeneration of endometrium. See, regeneration of endometrium partly occur in the follicular phase, whereas this endometrium will become more glandular in luteal phase. So we can go with the regeneration of the endometrium also here. Coming to the next, which of these is not an example of adaptive radiation? We know that adaptive radiation is what? There is an ancestral stock. Now this ancestral stock got adapted to different environmental conditions. So these organisms are adapted to different environmental conditions, so that what happened? Accordingly their body shape, size and different anatomical structure developed. So this is called as adaptive radiation. From one point they got diverged, right? So it is also what? An example of divergent evolution. So placental mammals, Darwin finches. Placental mammals means, say for example I can say man, cheetah, bat and dolphin, right? So these all are mammals. These all are mammals. But here what happened? Due to their adaptation to different environmental condition, their forelimbs are modified. So it is an example of what? Adaptive radiation. Darwin finches, that is also adaptive radiation. So what are Darwin finches? These are the birds, ancestral birds, those who flew from the South America to Galapagos Island. In Galapagos Island they found different type of food resources. So that is why what happened? They got adapted to different type of food. So those are Darwin finches. That is also an example of divergent evolution. Similarly, Australian marsupials, from the ancestral stock, different type of marsupials originated, right? So this is also an example of adaptive radiation. So the answer is third one, long neck giraffe. That is not an example of adaptive radiation. Next is a Hardy-Weinberg equilibrium question. In a population of 800 rabbit showing Hardy-Weinberg equilibrium, the frequency of recessive individuals was 0.16. What is the frequency of heterozygous individual? See, we know that frequency of recessive individual, that means what is given? Q square is given, that is 0.16. That means Q is 0.4. And we know P plus Q is equal to 1. So if Q is 0.4, then P should be 0.6. And what is the frequency of heterozygous? 2PQ. So 2 into 0.4 into 0.6, that is 0.48. So answer here is D. Next, identify the symptoms of pneumonia. In pneumonia, what happens? See, there is one more disease is there in NCERT along with this pneumonia, that is common cold, right? Or rhinitis. In rhinitis, what happened? Lungs will not be affected, only the respiratory tract will be affected. Whereas in pneumonia, what happened? Lungs will be affected. So lungs will be filled with the mucoid fluids. So that what happened? The person will be having difficulty in breathing. Okay. So here you can see option number D, nasal congestion and discharge, cough, sore throat and headache is given. There is no information about anything about the lungs. So we should eliminate this one. Student used to get confused with the D. High fever, weakness, stomach pain, this is no way related. Constipation, abdominal pain, cramp, blood clot is also not related. So answer is here second one, difficulty in breathing. Along with that, fever, chill, cough and headache will come. So whenever you will be having fever, automatically you will be having headache. Okay. So here the key word is what? Difficulty in breathing. So answer is B. Next, with respect to inbreeding, which among the following is not true? Not true. And with respect to inbreeding, so it helps in elimination of less desirable gene, that is true, very much true. Inbreeding decreases homozygosity, no. Inbreeding what? Increases homozygosity, that we call it as what? Pure lines. So inbreeding increases pure line, okay, or increases homozygosity and to help in establishing pure line. So this is wrong. Let us check other also. It helps to evolve a pure line, yes. It helps in accumulation of superior gene, yes, that is also true. So answer is B. Inbreeding increases homozygosity. In MOET, multiple ovulation embryo transfer technique, fertilized eggs are transferred into surrogate mother in which of the following stage? Surrogate mother it is talking about. It is not mentioning whether into the fallopian tube or into the uterus. So directly we can take 8 to 32 cell stage. Okay. So up to 8 cell stage, generally we used to transfer into the fallopian tube, and if it is more than 8 cell, then it is to the uterus. Okay. So here answer should be 8 to 32 cell stage. 32 cell means what? It is nothing but the morula stage. Next, Roquefort cheese is ripened by. Penicillium roqueforti. It is a fungus. The name of the fungus is Penicillium roqueforti. So it's a direct question. So our answer will be D. Now next, 4 students were assigned a science project to find out the pollution level of the lake in their surrounding. After analyzing the quality of water sample, the BOD value were found as follows. Which among the following water sample is highly polluted? So we know what is BOD. BOD is the biological oxygen demand. It is the amount of oxygen required by the microorganisms to degrade the organic pollutant present in 1 liter of sample, right? That means more organic waste, more biological demand. The organisms require more oxygen to degrade more pollutant, right? So more organic pollutant, more demand. So if the BOD value is more, that indicate more pollution. So among this 0.6, 0.16, 0.06 and here it is 6 mg. So here the BOD is what? Very high. So BOD is high means more pollution. Next, the toxic substance hemojoin responsible for high fever and chill is released in which of the following disease? That is direct question, malaria. So we know that malarial parasite infection, what happened? The parasitic stage that will eat away your hemojoin granules, eat away the hemoglobin. So when the hemoglobin will be eaten, then what happened? Hemojoin will be released. Okay. So that happens in malaria and this hemojoin granule cause fever. So this is the product of what? Degradation of hemoglobin. Degradation of hemoglobin. Next, which of these is not a method to make host cell competent to take up the DNA? So see here, to make the host cell competent, generally we are using what? Calcium chloride and heat. Okay, calcium chloride, actually the calcium ion along with the heat makes the cell competent to take up the DNA from the surrounding medium. But here what happened? This option is not there. But if you will see, the most irrelevant option is elution. Most irrelevant option is elution. So elution is what? Is the process by which the DNA bands are taken during the gel electrophoresis. Okay. So here we should go with the answer D. Next, select the correct statement from the following. Okay. The first step of PCR is heating, which is used to separate both the strands of gene of interest, seems to be true, because in PCR what happened? We have taken the double stranded DNA and we are heating at 95 degrees Celsius, so that what happened? The single stranded DNAs are produced. This hydrogen bond among them will break and this process is called as what? Denaturation. So this is true. Genetic engineering works only on animals and not yet successfully used in plant. No, there are many examples we can see where we successfully use recombinant DNA technology or genetic engineering in plants. DNA from one organism will not, it is actually printing error in your CET paper, it should read as bind. So DNA from one organism will not bind to the DNA of from other organism. No, it will bind. Okay, it will bind. So this statement is also wrong. There are no risk factor associated with recombinant DNA technology. No, recombinant DNA technology also associated with a lot of risk, right? Say for example, you can say the development of antibiotic resistant bacteria. Okay, so risk factors are associated. So answer here will be first one. Next, PCR is used for, PCR is what? Polymerase chain reaction, where we are producing multiple copies of a single piece of DNA, right? So that is nothing but what? DNA amplification. So here is also direct question. It is used for DNA amplification to form multiple copies of DNA. Find out the correct match. Okay. Filariasis is caused by common roundworm and main organ is small intestine. No. Actually, filaria is caused by what? Wuchereria bancrofti, where lymph nodes are affected. So that is small, it is not small intestine. Okay, so that disease is called as what? Elephantiasis. Ringworm caused by the fungus, and it affects skin, nail, hair, etc. Right? So this seems to be correct. Dysentery. Dysentery is caused by protozoa, correct, but it is not affecting liver. Typhoid is caused by bacteria, yes, Salmonella typhimurium, but it not affect the lungs. So answer is B. Next, match the following columns. Okay, Haemophilus influenzae, Entamoeba histolytica, Plasmodium falciparum, Wuchereria bancrofti, malignant malaria. This seems to be easy. First, see, you can see Plasmodium falciparum, it is causing what? Brain malaria. That is also called as malignant malaria. So how many options are reading 3 is P? 3 is P, only one option. Okay, let us check other also. Wuchereria bancrofti. Wuchereria bancrofti is what? Q. 4 is Q. Entamoeba histolytica, it causes amoebiasis. 2 is S. And Haemophilus influenzae cause pneumonia. So answer is D. Next, from the following tools or techniques of genetic engineering, identify those which are required for cloning a bacterial gene in an animal cells and choose the correct option. See, in that they have given around 8 options. What exactly it is asking? It is asking the technique used in genetic engineering where you are cloning bacterial gene in animal cell. That means this is a bacteria and this is an animal cell. So you are isolating a bacterial gene, okay. You are cutting the desired gene, you are putting in a suitable vector, and then you are putting inside the host cell, right. Now here what happened, definitely you are using RN, okay, and here definitely you are using ligase, finished. So 2 enzyme you have to use, one is restriction endonuclease, RN, and another one is ligase. So how many options are having 1 and 2? No need to check other options. Other option also you can go with 4, 6 and 8. Yes, 4, 6 and 8 also you can use, but in this way you can easily find out the answer. So here restriction endonuclease and ligase must be used. So only one option is reading 1 and 2. So here answer will be D. Along with, yes, microinjection, lysozyme and electrophoresis can be used. Next, flame cells present in the member of platyhelminthes are specialized to perform flame cells, solenocytes. These flame cells in Brachiostoma are also called as what? Solenocytes. So they are especially meant for what? Excretion. So excretion is also associated with osmoregulation. So here excretion always associated with what? Osmoregulation. So answer should be D. Next, the function of typhlosole. Typhlosole is what? These are the foldings on the inner wall of the body wall, you can see in the intestinal region, right. So that is why that intestine is called as what? Typhlosolar intestine. So this typhlosole, this folding actually increase the surface area for the absorption of food materials. So increase the effective area of absorption in intestine. So this is also straight question. Next, again match the column. Collagen, trypsin, insulin, antibody. So here we can see antibodies are what? They are a part of our immune system that fights with the antigens, so fight infectious agents. Insulin is a peptide hormone, trypsin is an enzyme, and collagen is the intercellular ground substance. So 4 should be P, 3 should be Q. Answer is first one, 3Q, 4P. 2 is the enzyme, 2 is the R, 1 is the S. So answer is A. Next, considering the stroke volume of an adult healthy being is 70 ml, identify the cardiac output in 1 hour from the following. So we know that cardiac output is what? Is the product of stroke volume and heart rate. And we know that cardiac output is what? 5 liter per minute. That means every minute 5 liter of blood is being pumped by your heart. So in 1 minute, if it is 5 liter, in 1 hour 5 into 60, 300. So how many options are there near 300? Answer is A, 302.4 liter per hour. Okay. Cardiac output is equal to stroke volume into heart rate. Okay. So when the stroke volume is 70 ml, heart rate is 72 times per minute, then cardiac output is around 5 liter. Okay. Next, function of contractile vacuole in amoeba. Contractile vacuole is for what? It will absorb excess amount of water and it will throw outside. That means it helps in osmoregulation, or it is having excretory function again. Yes, excretion and osmoregulation. Next, atrial natriuretic factor. Atrial natriuretic factor is actually produced from the wall of the atria to counter the RAS mechanism, right? So in the RAS mechanism, what happened? Your GFR will be restored, so that is why your blood pressure ultimately increase. So to increased blood pressure should be nullified, right? So that is why what happened? Atrial natriuretic factor will be produced, which act as vasodilator. It will dilate the blood vessel and it reduces your blood pressure. So it is not vasoconstrictor, hypertension inducer, promoter of renin-rho. It is acting against. So it check on renin angiotensin mechanism, RAS mechanism, antagonistic to RAS. So answer is B. Next, the vibration from the eardrum are transmitted through ear ossicle to what? Now this is the eardrum. Okay. So this is the ear, this is the auditory canal, and here is the eardrum, right? So here what happened? Ear ossicles will be there. Here one ossicle, here one ossicle, right? Malleus, incus and step. Step is attached to the cochlear region. So this region is what? This is oval window. Here you will be having a round window, right? So this is the oval window. Fenestra ovalis, and here will be the fenestra rotunda. So that means the wave of vibration will be picked up from the eardrum by the malleus. Malleus will transfer it into incus, incus will transfer into the step, and step, the foot process of the step bone act as a piston, and it is attached to the oval window. So when it will act as a piston, so inside it what happens? The endolimb and perilimb, whatever is there, they will pick up the vibration. So here it will be oval window, fenestra ovalis. Next, identify the structure. So you know that this one is a sinoatrial node, or P is SAN. Here it should be AVN, Q is AVN, and here it will be bundle of His. And here it will be interventricular septum. This is the interventricular septum. So P should be SAN, only one option, option number B. Next, in the female reproductive system, a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening is called as what? Clitoris. It is analogous to penis of male, clitoris. Answer is A. Next, consider two statements. Statement 1. The presence or absence of hymen is not a reliable indicator of virginity or sexual experience. This is a direct NCERT line. The statement is true. The sex of the fetus is determined by father and not by mother, because father only produce two type of sperm, X bearing sperm and Y bearing sperm, whereas mother will produce only the X bearing egg. Now, the sex of the fetus will be decided by which sperm actually fertilize the egg. So, this statement is also correct. So, both the statements are correct. Both the statements are correct. Statement 1 and statement 2 are correct. Okay. Next, the male sex accessory duct. Primary duct is what? Seminiferous tubule, where the sperms are being produced. Now, the sperms are being taken by certain ducts. So, those ducts are called as accessory ducts, or those who only transport the gamete, but they do not produce the gamete. Okay. So, retitestis, vasa efferentia, seminal vesicle is not a duct. Retitestis, vasa efferentia, epididymis, seminal vesicle is not a duct. Retitestis, vasa efferentia, epididymis, vasa deferentia. Okay. Retitestis, urethra is not. So, answer is C. Retitestis, vasa efferentia, epididymis and vas deferens. They are the accessory ducts, male accessory ducts. Next, with reference to human sperm, match the column. Okay. Acrosome is filled with the enzyme we know. There is a modification of Golgi that contains all sorts of enzyme that helps in breaking the wall of egg at the time of fertilization. So, 2 should be P. How many 2 is P? Only one option is there, 2 is P. Head contains haploid nucleus. Okay. Middle piece contains mitochondria, that we call it as a Nebenkorn, that provides energy for the sperm motility, and tail is sperm motility. Okay. So, answer is B. Okay. Thank you.